Thank you, Janice. Right, viruses are acellular microscopic life forms that can replicate only inside their host. So they are, by definition, obligate parasites. They consist of several parts. They can have single-stranded or double-stranded RNA or DNA genomes. And that genome is encapsulated by a protein code, which is called a capsid. Sometimes that capsid is enveloped by a lipid layer. For example, here in the top right-hand corner, you see a cartoon of an influenza virus. And this particular virus has a lipid envelope, but not all viruses have that. And sometimes viruses have a tail. And typically, we see that in many of the viruses that infect bacteria. They're called bacteriophages or phages. And many of those have a tail that are used to engage with their host and insert their genome into the host. The size of viral particles are very variable. They're mostly small, range between 20 nanometers and up to a micron, which is almost the size of bacterium, so actually relatively large. And they're morphologically very variable, and I'll give you some examples here. As with particle size, genome size is also extremely variable and ranges between about 2,000 bases and 2.5 million bases. There are two phases to the life cycle of a virus, to the replication cycle. All viruses have a lytic cycle, and some viruses also have a lysogenic phase. So when a virus connects with its host, it sort of it integrates with its membrane and inserts its genome into the host cell. And it then uses the host cell machinery to replicate its genome and make all of its particle components. These components get assembled, and then the virus lyses and kills its host. And those new viruses are released into the environment, and they can find another host and infect them. Some viruses are able, after they inserted their genome into the host cell, to integrate their genome into that of the host. HIV is an example of a virus that can do that. And this is called lysogeny. Often, those lysogenic viruses remain dormant within the host genome, and they are passed on maternally from one cell generation to the next, until a certain factor, often environmental factor, induces these viruses to enter the lytic cycle. And then it becomes an active infection again. Now, why are marine viruses important, and why did we decide to study viruses in corals? First of all, viruses are the most abundant biological entity in the ocean, and they infect all cellular life, from bacteria to whales. And this is a relatively new notion. Up until the 1990s, hardly any marine viral research was done because it was believed they hardly occurred in the sea. And there was a Norwegian group who published a Nature paper in 1989 demonstrating that they are actually highly abundant, 1 or 2 orders more abundant than cellular life. Viruses affect nutrient cycling because they can kill bacteria and algal blooms. Here's an image of a terminated bloom of Emiliania huxleyi in the North Sea. And from space, you can see all the calcium carbonate plates that are sort of floating around in the ocean. And this is done by viral lysis. Viruses can drive virulence of bacterial pathogen. Often viruses carry toxin genes. And if they insert their genome in that of their host, they can then turn a non-pathogenic bacterium into a pathogen. Viruses can also transfer genes between species and as such facilitate adaptation and niche expansion. They can also cause disease by killing host cells, but on the contrary, they can sometimes prevent disease by killing cellular pathogens. And again, in some instances, lysogenic viruses, once they insert themselves into their host, they can prevent infection by another virus in that host. So viruses can have a wide range of roles in almost all systems in the world. Now today, I'd like to talk about some of the work that we've been doing on coral-associated viruses, and mostly in the context of their role that they might have in disease, but in this case I will talk about bleaching. but also in terms of their possible roles in preventing disease. Now, the first note of viruses in cnidarian algal symbiosis was published in the early 1980s in the Hydra chlorella symbiosis. And after that, for about 2 decades, we heard nothing in the literature about cnidarian viruses until the early 2000s, when some reports started to come out, mostly based on microscopy, TEM, where people started to see viral particles associated with cultured Symbiodinium, anemones, and also coral samples. So the fact that there is some evidence that viruses are associated with Symbiodinium, and now also from more recent viral metagenomic data, led to the hypothesis that perhaps some instances of bleaching are due to lysis by a virus. From our own work, and I won't have time to go into detail, we have shown that based on metagenomes of coral samples, there are at least representatives of 34 of the 96 known viral families associated with corals. So it's a highly diverse group of life forms. Now, a second observation that suggested that perhaps some instances of bleaching may be due to viral lysis is that high temperature and UV exposure are known triggers to induce lysogenic viruses to enter the lytic cycle. And those are also 2 very important factors driving coral bleaching. And then thirdly, the host cellular pathways that are activated during viral infection are exactly the same as those known to be involved in coral bleaching, namely the production of reactive oxygen species, caspase activity, and programmed cell death. So we were interested to test this hypothesis, and to do so, we started to look for viruses that specifically infect Symbiodinium in Great Barrier Reef corals. And to do so, we mostly worked with unialgal cultures of Symbiodinium, because if we found eukaryotic viruses in those cultures, we could be sure that they were not infecting the coral host or any other eukaryotic organism associated with the coral holobiont. And we used a combination of methods. One, we looked at viral metagenomes in Symbiodinium. So basically what that means is that we isolate most of the viruses present in the algal culture, isolate their genomes, then sequence short bits across these genomes, and then use bioinformatic tools to reassemble genes and larger context and genomes of the viruses present. We also looked at transcriptomes of viral cultures as well as microscopy, TEM. And using these approaches, we found evidence for the presence of 3 different types of viruses in Symbiodinium. So the first one is a large double-stranded DNA virus that belongs to a group called nuclear cytoplasmic large DNA viruses. And most of the sequences we find in our transcriptomes and viromes belong to this group of viruses. We also find them quite commonly in metagenomes from the whole coral holobiont. Now, those NCLDVs are known to have a morphology that looks like this. It's called icosahedral. And in our Symbiodinium cultures, we find those kind of particles. And these particles are of a size that are known similar size to those of a virus that is known to infect and kill through lysis a free-living dinoflagellate Heterocapsa. So that is probably one virus that is definitely present in Symbiodinium. A second virus is a single-stranded RNA virus, and this particular virus is quite interesting. It's associated with the nucleus in Symbiodinium. So in our cultures, we tend to find a mix of healthy and disintegrating cells in control cultures, as well as cultures that we subject to UV or temperature stress. So here in panel A, we see a nucleus of a Symbiodinium cell with highly condensed chromosomes here. So this is a healthy cell. But we see a lot of cells that start to disintegrate. And in the nucleus, where the chromosomes are, we see the appearance of these filamentous particles. Sometimes the whole cell is filled with these particles, and we suspect that those are viral particles. Interestingly, when we look at the genomic sequence, the DNA sequences, these viruses are related to retroviruses, and retroviruses are known to integrate themselves into their host genome. We compared our data, our viral metagenome and transcriptome data with the Symbiodinium genome data. This particular strain has been sequenced by a Coral Genome Consortium called Refuge 2020. Some of you know about this. And we find some matches to retroviruses there as well. So it suggests this is another virus that is present in Symbiodinium. And interestingly, when we looked through the literature, there are two papers that published a proteome, one of a Symbiodinium enriched fraction from Stalophora, and another one is a proteome of the holobiont Acropora digidifera. And again, there are matches to retrovirus-like sequences in there. So quite strong support that this is another virus that infects Symbiodinium. And then thirdly, there's another single-stranded RNA virus. We find it's not highly abundant in the Symbiodinium viromes, but we do occasionally find it at very high abundance in coral viromes. Again, it's a virus that is previously only known from a free-living dinoflagellate, and it induces lysis and death in that virus. It has, again, this icosahedral morphology. It's a bit smaller than that large double-stranded DNA virus, and it matches the size of the virus in the free-living dinoflagellate. So 3 different viruses, we are pretty confident that they're present in Symbiodinium, that they infect Symbiodinium. Now, recently we have conducted a heat stress experiment on the main culture that we used in the experiments I've just described, which is this one here, the thermotolerant magnetic island Symbiodinium. It's a type C1. And we subjected these cultures to a control temperature of 27 and heat treatment of 32 degrees centigrade. And these are just 2 of the phenotypic measurements we took, maximum relative electron transport rate and the amount of reactive oxygen species produced, extracellularly produced, basically that leak out of the Symbiodinium cells. And you can see that at 32 degrees, there's no phenotypic evidence of heat stress after 15 days in this particular strain. Now we conduct, in the same experiment, we looked at a temperature-sensitive conspecific culture. This one comes from South Mole Island in the Whitsundays, and Emily Howells has previously shown that there is this difference in thermotolerance between these populations is due to local adaptation to local thermal regimes. And here we saw a clear phenotypic stress response. Now we also did a transcriptome on these cultures, and what was quite interesting is when we looked at the number of viral genes that were present in these transcriptomes, we found far more genes, viral genes, that had increased abundance at 9 and 13 days of heating compared to the thermotolerant magnetic island strain. And this higher viral activity or abundance in the heat-sensitive strain suggests that viruses may play a role in bleaching. It's still very early days, and we need to follow this through, but there is some hint that they may play a role. So where do we go from here? The viral metagenomics is quite labor-intensive. The bioinformatics is not straightforward. The databases are very poor for viral sequences, and it's fairly costly. So what we like to do is to develop PCR-based assays for these 3 viral groups, which will allow us to a bit more rapidly assess abundance in space and time, diversity, and so on, in a wide range of samples. And we have done this for one of these coral types, one of the RNA viruses. Now, viruses don't have a universal gene such as 16S or 18S ribosomal RNA in prokaryotes and eukaryotes, but luckily, within certain closely related groups of viruses, some genes are shared, and we can target those genes to develop similar assays. And for this particular single-stranded RNA virus, we targeted one of the structural genes, the major capsid protein gene, and we managed to design PCR primers, and we sequenced these amplicons, and they are specific, and we are analyzing them at the moment. So if we do this for all 3 groups of viruses, we can start testing this viral lysis of coral bleaching hypothesis a little bit further. Now this takes me to the second topic I'd like to talk about, and I won't talk for very long, I'm almost done. And so viruses, if viruses lyse a cellular pathogen, they can of course be beneficial to the host and perhaps cure disease. And one of our PhD students, Patrick Berger, is very interested in this topic, and he's studying black band disease. Black band disease is a microbial consortium dominated by cyanobacteria, as you can see here in this image. And it sort of grows over the coral surface, creates an anoxic sulfide-rich microenvironment, and kills the underlying coral tissue. And the question is phage therapy an option for control of this coral disease? We know that bacteriophages are often very specific to their host, and so they can possibly be used therapeutically to lyse and kill bacterial pathogens and perhaps cure and prevent disease. Now, first of all, Patrick cultured the dominant cyanobacterium from black band disease, and he sequenced its genome. And what he found is that this genome contains the CRISPR-Cas system. And this is an acquired immunity system that some bacteria have to defend themselves against viral infections. So what happens is once a virus inserts its genome into the cyanobacterial cell, parts of its genome are incorporated into this CRISPR-Cas system in the host genome. If there's a subsequent infection, then these sort of bits of sequence will recognize the virus genome, and that will then become marked and targeted for degradation, so the virus won't be able to kill the host. And so a lot of these sequences we found in this cyanobacterium genome matched to cyanophages, so phages that infect cyanobacteria. And that demonstrates that, yes, indeed, this dominant cyanobacterium in black band disease is targeted by viruses, so it warrants further research into phage therapy, which is the next thing he did. And so in this next step, he tried to actually culture the virus. And the way we go about it is that this line here is the control, and so we get these cyanobacteria to grow, and at the exponential growth phase, we infect them with viral concentrates. And those viral concentrates can come from a variety of environments. In this case, a lot of them came from seawater collected above black band disease in the field. And what you hope is that the specific virus for that cyanobacterium is present in your concentrate. So you have to use quite a lot of different concentrates. But at one stage he was lucky, and he saw that in the cultures that he inoculated with viral concentrate, there was a decrease in growth over time. When we looked at the cultures, this is the control and this is the inoculated culture, we see clear evidence of lysis. And this plot down here is a flow cytometer plot. And here within these red ovals is where we expect viral particles to appear. And you can see that hardly any viral particles present in the control, but lots of them in the inoculated sample. So he's repeated this 3 times, where he re-isolated viruses from the medium, then inoculated fresh cyanobacteria, and he found the same results. So we're pretty certain he's got a purified virus now, which he is sequencing to be absolutely sure. And then the next step will be to set up a phage therapy experiment and see if we can actually use this bacteriophage to cure the coral from black band disease. And so in summary, well, viruses have been found to play a range of important roles in every system they are studied. But our knowledge of marine viruses, and particularly viruses that are present on coral reefs, is very, very limited. And we've shown that at least 3 different types of viruses infect Symbiodinium. And our preliminary results suggest that there may be a role for viruses in some instances of coral bleaching. Again, it's very early days. Viral metagenomics can now be used to develop rapid PCR-based assays for further testing of this hypothesis. And hopefully this El Niño event will provide an opportunity to do so. And finally, the study of viruses may lead to small-scale solutions to treat coral disease. And I'd like to acknowledge everybody who's contributed to this work, which was funded by the ARC, AIMS, and AIMS at JSU. And thank you for listening.